All right, welcome everyone to this week's seminar of the Living Earth Collaborative and Washington University's Ecology, Evolution, and Population Biology program. Today, I have the pleasure of introducing Dr. Nicole Miller-Struttmann, the Lawrence L. Browning Jr. Professor at Webster University, who studies the evolution and conservation of plants and their pollinators. Dr. Miller-Struttmann is joining us today as an alumnus of Washington University, having earned her PhD through the EEPB program under the supervision of Dr. Peter Hoke. After her PhD, she went on to a postdoc with Dr. Candace Galen at the University of Missouri. Dr. Miller-Struttmann has an impressive record with research that has revealed phenomena ranging from climate-driven evolutionary reductions in bumblebee tongue length to how acoustic monitoring of buzzes produced during bee flight can predict pollination services. She has published in journals ranging from Science and Ecologia to those dedicated to scientific pedagogy. Dr. Miller-Struttmann also leads numerous community science initiatives, ranging from the St. Louis Bee Brigade to an initiative called Bee Clips that monitored bee activity during the 2017 total solar eclipse. In recognition of her contributions to community science and undergraduate education, she received the 2019 Science Educator Award from the St. Louis Academy of Sciences. One of Dr. Miller-Struttmann's current projects is Shutterbee, a community science program for tracking bee populations in the greater St. Louis area, which is the focus of her talk today. I'd like to remind everyone to please leave your questions for Dr. Miller-Struttmann in the live chat, and we will relay them to her at the end of her talk. And with that, I thank Dr. Miller-Struttmann for joining us, and we'll hand it over to her.
Thank you very much for having me. It's been a while since I've given one of these talks, ever since I was a graduate student at WashU. This is obviously a different venue for that talk, but it seems like we have been able to have a silver lining with Zoom and being able to share these talks online and through YouTube now, which is pretty exciting. So the presentation today focuses around Shutterbee, like Matt said, and this is really a collaborative effort. This is combining conservation scientists with behavioral psychologists, as well as outreach professionals, in order to sort of bridge what is often a gap between the conservation researchers and the people who are implementing conservation in the field, if you will. So to give a shout out to some of my collaborators right up front, Gerardo Camillo's lab at the St. Louis University, they are heavily involved in this program. You'll hear me talk a lot about the Bring Conservation Home program through the Audubon Society of St. Louis and the St. Louis Zoo, the Missouri Botanical Garden, as well as the Academy of Sciences St. Louis. And the work is supported by the Living Earth Collaborative and Webster University. So one of the things that is probably pretty obvious to most of us is that humans are having an impact on the globe, and that as our populations are increasing, so is our impact. So a recent study came out a couple of years ago now that estimated that 95% of the Earth is modified by humans. That's 95% of the land. Oceans are even higher. And more than half of that land has significant impacts. So if you want to take a look at this map of the globe and just sort of pick out your favorite city, if you know where it is on the map, of course, then you might notice that it's surrounded by red or maybe some deep orange or something that is implying a much larger impact by humans. So urban environments are incredibly important, and they are growing in terms of not only the number of people, but also in their range. And those tend to be areas where we have pretty heavy impacts on the environment. All of those decisions that we make when we are designing these urban environments create filters, ecological and then therefore evolutionary filters. So they influence what the ecology and the evolutionary biology of that system is. For instance, this is a figure from Shell et al. That came out last year that I thought has a really good way of demonstrating all of the different layers and the potential filters that could be involved in the impacts of urban environments on the ecology and evolution of organisms in them. So certainly we have Patterns that we've learned about for long periods of time, such as heat island effects. Obviously, if you have concrete or impervious surface, that's going to influence and change what organisms are able to do within an area or which organisms are able to persist there. We manipulate resources and change where they are, where they're available, both for organisms as well as for humans. And all of those things are driven by our social structures. So structures like racism, classism, or any other systemic biases that are built into our cultures. But these filters, if we can understand them and if we can understand where they're coming from, we can potentially weaken them. And that's a little bit about, I'll be coming back to that theme throughout the rest of this talk. So from my perspective, as somebody who is interested in plant pollinator interactions, and I've been working with bees over a decade now, is that we see patterns repeatedly. So for bees, even though you have or you can have pretty high diversity of bees in urban environments, it's not a perfect subsample of what would be available in the area. We tend to see more cavity nesting bees rather than ground nesting bees, in part because of that impervious surface and the structures that we're building in those environments. And we tend to have more generalized, less specialized species. So species that are more likely to have some sort of behavioral plasticity or ways of changing their behavior in a new environment, versus those that are a little bit more specialized, we're less likely to find in those environments. And those filters could be contributing to the global biodiversity declines that we're seeing. Like in many organisms, we're seeing this in insects as well. And a recent study came out demonstrating that it's not just declines in insect abundance. We have a fair amount of evidence from multiple locations throughout the globe that insect abundances are going down. But this paper really demonstrated that we're also seeing declines in biodiversity, the actual number of species that are collected within a given location. And so that's what this figure right here is demonstrating. It's just color-coded based off of decade. And these are called rarefaction curves. It's essentially a way of estimating how many species are likely to be in a location based off of how many samples you have. And the biggest draw point, or the biggest thing to focus on here, is that that purple line, which is the most recent decade, despite having a fair number of records, our number of species is significantly lower. And this is a pattern that they have seen, at least in that paper, in multiple locations, multiple continents. Some continents, it's a little bit stronger than other continents, but it is a pattern that is emerging. Why do we care so much about biodiversity? Well, for one, it's cool, right? I mean, we see all these beautiful, amazing organisms. We get attached to them. I'm known for saying that I think bees are cute and I think they're fuzzy and awesome and fun to learn about. But they also play important functional roles within the community. So when you have greater diversity in plant pollinator communities, you have greater robustness to change. You have redundancy that helps to stabilize the community. So this was demonstrated in a paper from last year from Udi et al., where they looked at communities of pollinators and how they interacted with plants. So that's the networks that I was referring to a minute ago. And what they found is that in locations, well, let me break this down a little bit more because there's a code here at the bottom. So at the bottom, it's broken down into three main categories: farmland, villages, and cities. And those villages are, this is a study from Europe, and those villages are sort of along the lines of a suburban, exurban kind of location. So in panel A, what you're seeing is the diversity of species within these locations, and this is for bees. And in the city, we see a much lower diversity of species. And with that, if you go to the last panel, we see lower network robustness. What does that mean exactly? Well, what we do to try to understand robustness of a network is we perturb it a little bit. We mess with it. We remove a species and see what happens to the structure of that network. So what happens in these city networks, if you remove a species, you get a much bigger change in the structure and potentially the long-term stability of that community. Now we know that organisms like bees can make different decisions. They can choose to switch and visit new flowers that they previously didn't. So there are behavioral mechanisms that can buffer communities from total collapse. However, there are also implications for what that might mean, what those changes might mean for long-term stability of the community. Our yards, parks, farms, public lands, as well as private lands could be part of the solution. And there's been a big push over several years now to really get ourselves thinking about land as a resource, not just for us, but for these organisms that we're trying to preserve. And this is something that has been spearheaded by Doug Talmy. He's been doing all kinds of outreach and written many books for the public sphere, trying to connect people with thinking about their yards as potentially natural lands, right, or places for wildlife. And he makes the point that lawns, which from an insect's perspective are really almost deserts, at least for the pollinators that my bias is towards. But if you look at lawns, and this estimate's actually quite old at this point, it's 15 years old. But in 2005, the estimate was 128,000 kilometers were just straight up lawn. And lawn is obviously not a natural habitat for this region or any region really, although grasslands certainly are. But if we were to convert these lawns to something that was a little bit more naturalized, a little bit wilder, we might be able to build in that buffer within urban systems. And there's been some evidence that supports that idea is a possibility. So there was a study that came out a few years ago now that looked at allotments, and this is in the UK, looking at different kinds of allotments and seeing where pollinators were most abundant and where the diversity was highest. Probably not too surprising, that was in gardens, because that's where you're going to see a lot of the highest diversity of flowering plants. However, those gardens do not necessarily reach the diversity as you would get in natural lands. So there's been a big push to engage people in their backyards through backyard conservation programs to promote changes that might actually help us weaken urbanization filters. One that is local is the Bring Conservation Home program. And this is a really cool program where individual landowners can apply to participate, and then they get individualized feedback for their location on how they can modify that landscape to support biodiversity. So they then get ranked based off of, or certified based off of how many of the different layers they've actually hit. So there's 3 layers: silver, gold, and platinum. And those rankings are based off of how many invasive species they have, what the complexity of the habitat is, whether or not there are water sources, and a myriad of multiple different factors that could help to support biodiversity. And so when they enroll in this program, they get a little bit of individualized feedback. And as they go through the program, they get reassessed to see how their site ranks in terms of conservation potential. However, it hasn't really been assessed. We don't know for sure. And most of these, or many of these backyard conservation programs haven't been assessed. So we don't know exactly how effective they are at creating the changes that they're intent on creating. And if anybody has ever tried to change one of a bad behavior of their own, we know that changing behaviors or changing culture is really challenging, particularly when we aren't considering the factors that actually change behaviors. I'll get a little bit more about that a little bit later in the study, sort of, or in the seminar, sort of priming you for this idea about what influences us, what changes our behaviors. And it's not easy to change those behaviors. But one opportunity to do this, to not only learn about what conservation steps really can help support biodiversity, but also to engage with community, or with the community, is through community science. So to invite people to help us learn about conservation, to help us study these behaviors in terms of land management decisions and how that influences bee diversity in this case. But at the same time, we're potentially building agency, if depending on how we structure the program, we can build agency within that group to actually change some decision making, have conversations going back and forth, and to really learn from each other, have true collaborations, because community science works best when they are more collaborative and when everybody feels like they are putting something in and getting something out. So that leads me to the Shutterbee program and the two overarching research questions that I am going to talk about today. So the first is, can backyard conservation programs weaken urban ecological filters of bee diversity and bee behavior? So this is the ecological question. And this has multiple subcomponents to it, as does question 2. So I am really giving you the broad overview of the two main pieces of the project. The second question deals with the human behavioral aspect. So does engagement in community science promote changes in conservation behaviors? So I'm going to take those one at a time. And we'll start with the first one, looking at whether or not backyard conservation programs weaken ecological filters. So for this, essentially what we're trying to understand is whether or not we can move, say this garden right here, these are gardens, this is species richness in gardens, this is species richness in semi-natural habitats. So in this case, the gardens in the dark spots are, while they do have similar responses to urbanization as the semi-natural areas, they have significantly lower species richness. So backyard conservation programs have the potential to try to inch that upward and perhaps flatten that line. So the Bring Conservation Home program has been incredibly supportive of these collaborations, and we've been working with numerous individuals who are enrolled in the program. This program has over 1,000 locations, well over 1,000 at this point, that they have provided people with feedback and have given them their individualized sort of trajectory or things that they could do to improve their yards. And we, while not all 1,000 of those are involved in the Shutterbee program, we did invite all of those individuals to be part of the Shutterbee program. So to give you a sense for whether or not there's some support for this idea that our behavioral decisions could influence bees, this is work from Nina Fogel at St. Louis University, where she went out and looked at the floral richness in BCH houses and then their neighbors. So high ranking conservation houses are those that had gold or platinum certification levels. And then low are those that had silver. And then unranked were locations that were enrolled in the program but had not yet received their feedback. And so what you'll see is in the high species or in the high conservation ranking locations, species richness is significantly higher. It's a little bit higher in the low conservation ranking sites as well, and it is not different in the unranked houses. So that is suggesting that with increased plant species richness, we could potentially have increased bee species richness. We might see changes in behavior, such as expansion in floral foraging, and that could influence the networks, both the stability of the networks and how robust they are to perturbations. So we asked folks who were in the BCH program, as well as the general public, to start to observe and photograph the bees in their own backyards in order to do this. And I guess it was a little bit of serendipity, but we started this program about 9 months ago. We have been planning for it for longer than that, but it started right in the middle of the pandemic. So we ended up having an incredible outpouring of interest in this program. And we ended up with 168 people who went through the full training. We had even more than that who were interested in the program, but for whatever reason did not fulfill, right, the full training. And this has been absolutely incredible. And we had a lot of folks, I think, that we wouldn't have been able to engage in any other time because people were home so much. And so that was a really exciting, but also a little bit daunting because we weren't planning on this project being as big as it was, but we seized on the moment and we had the support to do so. And it's been a pretty exciting, fun year. And I'm going to tell you a little bit about what we have found so far through this program. But I wanted to tell you a little bit about how we train our community scientists. So you get a sense for, we're not just setting people loose to take photographs in their backyards as willy-nilly. We really wanted to provide them with guidelines and supports in order to do it in a truly scientific way. But our first meeting didn't touch on that. Our first meeting was really about, our first training was really about building confidence and establishing community. So we all came together and we talked a bit about bee declines. What are the factors influencing those declines? What is a bee? It's way more than honeybees, right? Most people think of honeybees when they are thinking of a bee. That's the image that pops to mind, maybe a bumblebee. And so we really tried to get this idea that diversity of bees is way higher than the average person thinks it is. And to get them really starting to think about the potential for their own yard, the potential for who might be in their yard. The second workshop is where we actually got into the experimental design. And so these were smaller group trainings where we had everybody create a map of their yard to decide how they were going to move through their yard. We tiered the duration of the surveys based off of the side of the yard. So they either observed for 20 minutes or for 30 minutes. And we talked a lot about standardizing these protocols. So doing it at the same time of the day, when it is sunny or partly cloudy, because if it's very cloudy, that changes the bees' behavior. When it is warm and the bees are active, but maybe not so hot that the bees become less active. And to get them thinking about all of the bees that are in their yard, not just the big ones, but the little ones that people think often are flies or are gnats, but they are in fact bees. Then there was a third training component. And this is where we sent individualized feedback after people did their first surveys. And so you can imagine that for 168 people to provide individualized feedback, we really needed a lot of support. And I really want to give a huge round of applause to Emily Burley, Cody Dinkins, and Jessica Clonis, because they really were the front of this, the sort of where the boots meet the ground, if you will, in terms of engaging with individuals and giving them feedback on their photographs, on their survey technique, and really helping to empower everybody to be able to conduct these surveys in their yards. And then as they uploaded their photographs into iNaturalist, we all, the three undergraduate students, as well as Nina Fogel, tried to identify those species on iNaturalist. The really cool thing about working with iNaturalist, so it's a platform that's built for community science. And there are a lot of people who are active on there, regardless of whether or not they're involved in a specific program. And there are some pretty stellar hymenopterists on there. So we would get bee identifications in the middle of the night from some collaborators, wherever they were in the world. For instance, John Asher, who is a famous hymenopterist, feels like he's on iNaturalist all the time, which is really exciting because that's fun for us, that's fun for the students, that's fun for the community scientists to actually engage in a community that is outside of just within the St. Louis area. So in 2020, we ended up having about 8,209 observations that were recorded, and about 89% of those we were able to get to genus, and some of them to species. Some of these bees are so small that you really need a microscope in order to get to species, whereas other bees like bumblebees, it's actually quite possible to get to species level using photographs that you can take while they're moving, and with a phone. Now there's a caveat there. We can do that here. That's actually pretty challenging to do in some parts of the country where there's more morphological overlap. But here we're in a place where we can really tell some of these species apart, which is pretty exciting. So we've spent the last couple months culling these data and analyzing them. And in order to institute any new data collection technique, right? You want to know how it performs in comparison to what the traditional processes are. And so for that, we've been working a lot with Nina Fogel. She's a heavy lifter in the Shutterbee program. And she did her own nettings at 65 houses throughout the St. Louis region and collected 3,500 bees. Now, all of these bees can be identified all the way down to species, right? We can get a much higher taxonomic resolution, but it's very labor intensive. And what we wanted to know is, can we get similar patterns, or do we see similar patterns from photographs as we do from this really high quality netting? And interestingly, oh, sorry, I was going to go right to the results and forgot about my map here to show you where these locations were. So we had 37 locations, those are the blue squares, where both Nina was netting and we had participants actually photographing at those locations. Then we had the general community who was not part of Nina's program or part of BCH. We had 116 of those individuals. And then we had individuals who were part of Shutterbee, part of BCH, but not in Nina's sampling protocol, and that was 27. So we're going to be focusing on that middle blue, the blue squares here. Those are the locations where she was able to resample. So these are called rarefaction curves, and it basically just shows you, given an amount of effort, what is the estimate for the species that is in this area. And when we look at a very coarse grain, so this is across all of the locations, we see that for regional richness, we get a decent estimate for what Nina was finding. If hers maxed out around 25, ours maxed out around 20, which is not incredibly high, or it's not perfect, I should say. However, it is close. And when we think about many of the other techniques that are used for sampling bees, for instance, passive measures that are commonly used, like pan traps, netting documents the greatest amount of diversity. So if we are a little bit below netting, but still performing pretty well, comparatively speaking, then we're happy with that. However, when we look in a little bit more closely at the data and we look location by location, we see a lot of variation. So on the x-axis here is the rank. Each location is ranked based off of its similarity to Nina's netting. So the closer those are in terms of species richness, the higher the number. And you'll probably notice that it's maybe not quite as high as we would like. There's a significant amount of variation all the way down to 0.2, which is quite low. However, perhaps not surprisingly, this top line here, which is red, that is for the larger bees. So the larger bees were able to track better than these smaller bees. And you'll see for the smaller bees, it really does get quite low. So we really need to be careful about which species we actually can infer real ecological processes with and which we cannot. So for the big bees, we have a similarity on average of about 0.66. We're still hoping to improve that going forward, but that is certainly a very promising result. And then for those smaller bees, we really need to tease apart why they're not being picked up. Do people not know how to look for them or that they're even there? Are they having difficulty finding them? Are they having difficulty photographing them? So that's something that we're going to explore a little bit more this year. Now for those bigger bees, and I should have mentioned this, but this is at the genus level. So there are some species that we can reliably pick up on regularly, and those are even the biggest bees. So our bumblebees, Xylocopa, the carpenter bees, honeybees, as well as some other pretty charismatic larger species. So for those species, we feel comfortable moving forward and looking at things like visitation patterns. And that's what students in my lab have been starting to do. So Emily Burley, who I mentioned earlier, she was one of our Shutterbee aficionados, our ambassadors. She spent this fall conducting a senior thesis looking at whether or not bee behavior has changed, or is different, I should say, based off of the conservation status and along an urbanization gradient. So the figure that I'm showing you here is for honeybees. Honeybees, while not native, are quite common. They've naturalized within the area, as well as many people have hives. So it's not too surprising that we found them regularly enough to do this kind of study with. And she found something really interesting. When we have high or low conservation rank, so those are these two middle box and whiskers in the middle here, we have much greater foraging generalization for those individuals. So individuals that are found in high species or high conservation rank or low conservation rank yards. Whereas when we're in unranked yards, it's lower, although quite variable. So you'll notice the variation around that is much higher than in the conservation ranked. And part of that is likely because some of those unranked houses maybe do have a fair amount of floral diversity, but it doesn't reach potentially the height of our hard conservation areas. Now, interestingly enough, the other 4 species that she looked at, so Bombus gricicolis, Bombus impatiens, Bombus bimaculatus, and Xylocopa virginica, exhibited no changes in behavior. So none of the native bees that she tested actually seemed to respond to conservation status or to urbanization levels. There's a little bit of nuance there that I don't have time to get into today, but it appears that at least for this year at these locations, there wasn't too much of a change in how generalized they were. But this doesn't tell us who they're visiting, if they're overlapping in their niches, if they're visiting more of the same things, less of the same things. So there's still a lot more that we need to explore here. So next steps for question 1 are to really look at that smaller bee documentation and try to address what barriers are keeping those sightings lower than what we find via netting. And then also to go to that next level, the community level for the large bees, to look at their behavioral shifts to see whether or not there are changes in the structure of those networks. Now on to question 2, which is really exciting to me. This is a true collaboration. I am by no means a behavioral psychologist, nor do I do research in this historically. However, I have an interest in it, and I've been interested in outreach and the effects of outreach on the community for a long time, but we definitely wanted to bring in the experts. So this is a collaboration with folks at the St. Louis Zoo, including Aaron Tate and Amy Niblowski. And so what's amazing and fun about this is that we come from very different perspectives, yet at the same point, they have insights that relate directly to what we're trying to do with our conservation research. And so Briana Robles, who is a student in my lab this semester, is collaborating with Aaron and Amy to address this question of whether or not does engagement in community science promote change in conservation behaviors. So if you're like me, you may have limited exposure or experience with behavioral psychology and what influences how the decisions that we make. So I'm going to give a little bit of background on that. Now, community science work there's been an interest in how it might influence behaviors or people's connections to nature. And this was a survey that was conducted 3 years ago now, or published 3 years ago now, that demonstrated that nature-based citizen science projects enhance time spent in nature, or personal investment, observational skills, excuse me, and that those things influence their experiences of nature as well as that's influenced by the availability of nature, right? So we have multiple aspects that are influencing how individuals are experiencing nature. And I'm going to highlight this one right here, personal investment. So this is the perception of some sort of value and possibly that value is coming from nature and affecting those individuals. So that actually goes up through community science participation. And all of those things can lead to an emotional connection, a sense of commitment, but we don't really know what that means for behaviors, particularly high cost behaviors that are not easy to implement, or that maybe go against the grain, the cultural grain. And while we have a fair amount of studies that are out there exploring how community science can influence domain specific attitudes. So that means if you're in a project that is about bees, well, you learn about bees, you have an appreciation for bees, but do we get any cross-domain spillover, if we will, which is probably needed in order to make larger scale conservation, big conservation decisions? And we don't have an understanding of that in large part, according to people who are better, who are experts, not I, is because it's not really grounded in behavioral theory. So we, for this project, are using the theory of planned behavior. And that theory essentially says that there are three main components that influence whether or not somebody is going to engage in a behavior. There are behavioral beliefs, normative beliefs, and control beliefs. So I'll take these from the bottom up in this case. Control I think of as agency. How do you feel about your control of those behaviors? Do you feel like you can change those behaviors and actually make that change happen, right? Behavioral beliefs, a flip to the red on top, is that you can actually sort of predict that as you do that behavior, you're going to get some sort of change or consequence back from it, right? So it's your attitude towards it and your belief that something is going to come from it. The normative beliefs are about how people that are important to us, so it's not always all of society, but the people that we care about, what they think about these behaviors. And all of those things influence the likelihood that we are to engage in a behavior. Now, this theory was developed in part, or at least it's been used a lot in human health behaviors. So for the cessation of smoking and things like that, behaviors that we know are challenging to change. And it has been effective in doing that when these different components are addressed. So let's return to the Shutterbee program and look a little bit into where we might be addressing either control, normatives, or behavioral beliefs and how that could result in changes in behavior. So we start with an introduction into why we personally care and why they might care about bees, what's influencing their declines, right? This is more about a background thing, but it does sort of influence, or is one component, if we are important enough within their domain of thinking, it could influence normative beliefs. We also do a fair amount of talking about and support in how people can make different decisions for their yards in order to support diversity. So that gets into control. Here are things that you can actually do on a small local scale that could potentially have impacts for bees. So reducing pesticide use, being a lazy mower, gardening with native plants, providing nesting. These are all behaviors that can be taken that are relatively low impact, or I should say low effort compared to others, especially the lazy mower one. Everybody always loves that one. And so it's essentially saying these are things that you have power over. And we also have developed curriculum that really helps to engage people with the organisms, to learn about those organisms and what those organisms need and what they are. And this is another amazingly fun part of this collaboration, working with Mike Dawson and James Valpell. This is the best picture I could find of Mike Dawson with his Optivisor on and everything. And James Valpell, who's at the Missouri Botanical Garden. And we're developing curriculum both for within Shutterbee, but then also to take out to teachers to engage younger students in the bees in their yard. But engaging them in a meaningful way and helping them learn how to use keys to guide, to identify bees in their own backyard, etc. So in addition to our trainings, we also regularly communicate with our participants through social media. If that is something that they are interested in, they can join us on Instagram, Facebook, or Twitter. And we share conservation recommendations, information about natural history. And we also try to highlight some latest science, some latest conservation science. So and this is about getting at those behavioral beliefs, that something that changes that we're making might actually have an impact on these systems. And so some of this conservation I'll take one example. Sorry, the lazy mower one. The lazy mower thing doesn't just come from somebody mowing their front yard and noticing all the flowers were getting cut off. There's science behind that, where a group did a series of mowing treatments, and they determined that in that location, when you mowed every other week, bee diversity increased dramatically. So we try to essentially try to provide evidence for the things that we're recommending as a way to show that it can be not exactly predictive, but that there is support for these things really making an impact. We also send out bi-weekly, well, it was monthly, now it is bi-weekly bulletins that share some of that same information as well as some additional information. We talk about the history of bee science and some other really fun things. And we have some just engagement through challenges or games in order to make it enjoyable as well. So in order to test whether or not Shutterbee is influencing or participation in this community science program is influencing behaviors, we of course have to have control groups that are either not in Shutterbee or not in both Shutterbee and the Bring Conservation Home program. So we sent out survey requests, behavioral survey requests, I'll show one of them in a second, to people who are in the Shutterbee program and in the Bring Conservation Home program. We have 43 responses from that. People who are in the Shutterbee program, but not in Bring Conservation Home, we have 59 survey respondents from that. And then those that were in Bring Conservation Home, but not in Shutterbee. So here we're trying to tease apart Shutterbee per se and the Bring Conservation Home program. So what we're going to do is compare their responses to surveys that are conducted or that are meant to quantify their conservation readiness or behaviors before and after their experience in Shutterbee. One such scale that we're using, it's a published scale that has been validated and has been used for at least a decade. And this is the environmental identity scale. One of the things that influences behaviors and influences whether or not people are likely to engage in in conservation behaviors is how they identify with the environment. So what is their environmental identity scale? So it's a series of Likert style questions where individuals will answer whether or not, or they'll rank, I guess you could say, whether or not they feel that a statement is true of them or not. And what these questions get at are multiple parts of the environmental identity. So it ends up being you sum all those things up, and that gives you a sense for that individual in comparison to other individuals, how strong is their environmental identity? And potentially has that shifted through time, through their experiences in these programs. So unfortunately, I don't have preliminary analyses for this experiment yet, or this part of the study yet, because we are just getting into these data. They were collected at the end of the foraging season. But Briana is working hard getting these data together in R and working with Amy and Aaron to analyze those data. So she'll be working on that all of this semester, and we should have some interesting preliminary results by the end of the semester when she presents her talk for her senior thesis. So that's our next major step. And then we are also going to, of course, continue to track individuals who are involved in the Shutterbee program, as well as any new individuals who join the program. So our take homes from my talk today, as I see them, are that community science can offer opportunities for achieving conservation and research goals. However, successful community science really requires significant training and input and collaborative efforts and collaborative approaches with those who are involved. So I used to sort of, the ideal of having a community science project, you hear some people speak of it as a 2-for-1, right? It's really not a 2-for-1, it's a 2-for-2. So you don't get, double the data for half the work or whatever. It still takes a significant amount of work, but in my opinion, it's incredibly worth it. And it's allowing us to achieve multiple goals simultaneously by engaging folks in our research. And then finally, that promoting changes in conservation behaviors, we know it's going to be challenging. However, if we ground that in theory, in behavioral theory, then that gives us an avenue for creating either changes in individuals' behaviors in some way. So with that, I am going to do a quick little solicitation. If you know somebody who would be interested in being involved in this program, please do share this with them. They can either email us at shutterbee@webster.edu, or they can check out our website. And we actually are going to be soliciting people to take photographs in their yard, but we're also looking for people to help us learn to identify the common bee species. Because one thing we found is that it takes a while to identify 8,200 some odd bees, and in a way where we have at least 2 of us check each one of those identifications so that we know that it is accurate. So with that, I am ending up here with about 10 minutes for questions, which I would love to take. So I'd like to say thank you very much for inviting me, and I'll field any questions you have.
All right, thank you so much, Dr. Miller-Strutmann. Really, really fascinating work. So I'd like to remind everybody to please leave your questions in the live chat for Dr. Miller-Strutmann, and we will relay them to her. As we're waiting for questions to come in, I may start with one of mine that occurred to me while you were talking about your student Emily measuring foraging diversity. What method did she use? Did she measure foraging diversity at the individual level, the species, the genus level?
So what we did for this, we were looking at individual species, the species that were represented in multiple habitats. So we had sort of a cutoff for how many individuals had to be collected at a given site in order to get a true estimate for their foraging richness, right? So it would be at a location, we would get an estimate for that species. And then we would compare that across using each of those locations as a sample.
Interesting, thank you. So I know you mentioned that it might take a while to fully elaborate, but I'm curious why you think you didn't see an effect with bumblebees or honeybees.
Yeah, so we saw it with honeybees and not with any of the native bumblebees, which was really surprising. All of these species that we were looking at are considered generalists. She had a suite of specialist species that she was planning on looking at, and they were here, but not in high enough abundance to actually do statistical analyses with, which is a little telling right there. However, when we think about, and I'm sorry, I lost my train of thought. I got so interested in these rare species that aren't existing. So can you just repeat the last part of that sentence? I'm so sorry.
Sorry, yeah. So why
Oh,
do you
right,
think you didn't see the effect on
right.
foraging diversity with bumblebees?
Yeah, apologies for that. So I think it probably in part has to do with the fact that they already are so generalized. Maybe they are maxing out what they can possibly visit just because there isn't much available. They have to visit everything they can. They don't get to be choosy, right? So that's the flip side of things. But we're not seeing that they're getting less specialized or more, you know what I mean? So we're not seeing any patterns. And it really was surprising just because you would expect if there's a greater diversity of flowers, maybe they're visiting more things. But there's also a composition issue, right? So what Emily was looking at was really strictly speaking number of species, not the identity of those species. So it could be that in these high conservation locations, they are actually visiting not a wider breadth of things, but different things, and potentially things that are um, maybe more nutritious or have more nectar. You know, we don't have that kind of scale of information, but that'd be an interesting thing to explore going forward to see if they're actually foraging from a different subset of the plants.
Thank you. Yeah, that'd be fascinating to look at.
All right. Yeah, thank you so much. That's an excellent talk. Really excited. I actually hope to join in on one of these IDing
Great.
uh, situations. It seems like, yeah, it just seems awesome to be able to ID all the different bees here. So I have a bunch of questions, but I guess my main question is, so I own a house in South City, and I've been planting a bunch of native plants, all focused on different host plants for moths and butterflies, because that's more of my interest. But at the same time, I also very much care about bees. Do you have like two or three that you would say, look, everyone should have this flower in their yard to increase native bees and just overall maybe the insect fauna? Do you have like two or three recommendations?
Um, I am partial here, but I'm going to say echinacea. I'm a big fan of the local echinacea. They host specialist species as well as a lot of generalist species. They're bees like them, butterflies like them. They don't produce a ton of nectar, but they do. The pollen is pretty. Um, they get jazzed about the pollen. This is another bee bias, so sorry here, but I'm going to say Monarda because a couple of the species that we don't have as frequently as we should really love Monarda, but that one has a lot of nectar and pollen, and it's one that's favorited by Bombus auricomis and Pennsylvanicus, which are two that are pretty steady in the area, but certainly perhaps not as common as they used to be. Pycnanthemum is another one that's in the mint family, and that one, lots of small bees like that one, and it's also the leaves are used for some of the cavity nesting bees and things like that. So that's another good one. Several in the mint family.
Cool.
I give you three.
Yeah, no, that's great.
I can also shoot you whole lists. I mean, we got like I could go on for hours.
Yeah, so my follow up to that is like, where I assume if I go to the website, I would be able to find a bunch of species lists or where I can buy these plants, I assume.
Yeah, we don't list locations to buy, but we have a whole bunch of, I mean, there's amazing resources out there. These are not resources that I have built, but we are accumulating them and trying to put them on the website so that people who are at least within our program have a place to go to learn to make those decisions and have a place for when they're making those decisions, right? So yeah, there's all kinds of information in there. I have my own species lists, but there's a bunch of species lists. There's been whole books on this, right? So Heather Holmes has several books and on plants that are good for pollinators in general, where they have all kinds of fantastic lists.
Awesome. Excellent. And really quick, just real quick question. So, mowing laws in St. Louis, like, do you have some of your participants get in trouble and get notifications? I mean, one time I didn't mow my grass for like 2 weeks and I got a notification. So, and it's funny too, because there's tons of like empty lots that, have terrible lawn, what do I want to say, being manicured. So yeah, is there any like loophole where I can say, oh, this is bee habitat?
That's going to depend on the municipality, and that's going to depend on their feelings about it. There have been some publicized heavy discussions, I'll say heated discussions between homeowners for like in Maplewood. There was somebody who was trying to convert her yard to a more natural look, and that was not perceived well by some of her neighbors. And so we haven't heard of anybody dealing with this in our program, but it is a general problem. But it's about a cultural shift. This isn't an easy cultural shift. This is something that we have as it's a hangover from our European roots that, having these lawns is a status thing. Makes it sound like, oh, you're just out for the status. But with that comes comfort, right? It's something we're familiar with. It feels controlled. It's something that we can have a handle on. And so those kinds of cultural shifts are not going to be easy. And I think I'm always happy if somebody does come in, have that issue, I'm happy to come speak to anybody who wants to hear more about why pollinators should be, part of our backyards.
Excellent. Thank you.
All right, we have a couple of questions that have come in from people in the audience. Tom Sullivan wonders if you can speak to the type of cameras used by participants in Shutterbee and whether there is a learning curve in participants improving their photographic skills. Do you know the typical amount of time it takes participants to improve their photography?
So a couple questions within there. It varies considerably, both of those things, what they use as well as the skill of the photographer. We have some spectacular photographers who are just using these amazingly wonderful cameras, and they can take these just absolutely incredibly beautiful photographs. We don't necessarily need the beautiful photographs, but it does help often because they're nice and in focus and up close and things like that. Most people, though, are using their phones because that's what they are familiar with using. And what we have found is when people try to use something that is not familiar to them, it's actually lower quality. So we always encourage them to, whatever they feel comfortable with, to use that. And then we also encourage them to practice a lot before they get started. Now, we haven't done a statistical analysis of this, but there was a student who was in the collaborative master's program at the Missouri Botanical Garden, and she looked at the quality of the photos through time for a study that she was conducting for a class. And she said it really didn't seem to matter all that much. They might have gotten a little bit clearer through time, but we were still able to get them to genus. So I think the amount of emphasis that we put on practicing early probably gets them about as far as they're going to get until we have maybe some other kind of intervention or some other kind of, all right, let's sit down and really work on this hand to hand, together physically with your camera and help you get the right angle and work with light and work with speed and all that kind of stuff.
All right, we have another question. I think this will be our last one. So Michael Moore asks, do you have any sense how the refraction curve of non-Shutterbee participants taking bee pics compares to the trained Shutterbee participants?
So do we have, let me just state that back to make sure I'm getting the question right. Do we know what the rarefaction curve looks like for non-Shutterbee participants? Is that basically the question?
Yes. Yes. I wanted to make sure I wasn't muted. Yes, that's correct.
No, we don't, because we were looking at the individuals that we trained. So
Right.
we don't know what it is like for any individual who hasn't gone through this process at all. So we don't know, in other words, we don't know the effectiveness of our training, right? We don't know how far we were able to take individuals. We just assumed that we would at least need to give them some training to get them to that point. There is a way that we could potentially do that. So iNaturalist is an open source data set. And so we could potentially, we wouldn't know necessarily people's photographic histories, but we could look at their photographs on iNaturalist through time and just see whether or not their quality improves. There'd be so many factors that wouldn't be controlled for that, though, that I think that that would be really challenging. If we were really to test that, we'd have to just invite people and give them no support. And I don't know that I'm entirely comfortable doing that, because it's a lot of work to go through these, to photograph these in your yard and submit it.
Yeah, it would be interesting, like, moving forward, if you did just give people like random photographs at first, right before you train them, and just to see. Michael Moore is very heavily involved in iNaturalist, so I wonder what he has to say about that as well. And hopefully, yeah, you probably have had conversations on this, but hopefully you guys can follow up in future. Well, thank you so much. I'll give it back to Matt to close this out.
Great. Well, thank you again for joining us today, Dr. Miller-Strutmann. And I suppose we'll finish out with a comment here from Susanna Eisenbraun, who says, as a participant, I can say I learned a lot from your ongoing training and encouragement. So thank you again. And we thank everybody in the audience for tuning in.
Thank you very much.